welcome everyone. As I said before, if you have questions, I will also be asking you a few questions in the webinar just to make sure you understand the topic. We'll be using this Q&A dialogue here, and the reason we use that as opposed to the chat is I think it's recorded more easily and can be viewed later. I did paste a few things in the chat, which I hope you can access. The first two are papers by Dave LaBelle and Jones in 2013 and 2017, and they just provide you with some more background information on the topic. I read a lot, and these two papers I found to be very useful in understanding how remote sensing has evolved to estimate crop yield and used in agricultural monitoring systems platforms. And then finally, this is recorded. It's a 2-hour webinar. It's available for free on Vimeo. It'll be much of what I'm talking about today. The benefit of watching it now here is you can talk to me, ask me questions. I've been in the field for a long time, so it's more interactive. But you know, we know how technology is these days. If there's a problem with your internet or something goes wrong, you can always refer back to this Vimeo. That way you still get the information. Yeah, so let's see here. There's a couple more questions. Okay, I don't know NASA Power Data Viewer. I don't have experience with that. Okay. Let's see. It looks like, all right. See if I can paste this information in the Q&A. All right. Okay, I've answered. Okay. I've now answered in the Q&A. Hopefully you can see there's the two papers in the webinar recording. Yeah, okay. Any other questions so far? Once I start, I'll answer questions, but it's a bit hard for me to give the presentation and answer questions at the same time. So I'll answer you from time to time. You can also, I believe, ask questions orally, you know, so just turn on your microphone, not a problem. So let's start. My name is Michael Marshall. I'm an assistant professor in the Department of Natural Resources at ITC, University of Twente. My background is in remote sensing, both hyperspectral, multispectral broadband, sort of field level, but also large area, crop yield estimation, crop water use, evapotranspiration. So I have years of experience with this, and I've lived in East Africa for about 20 years, mainly Tanzania, Uganda, Kenya. Uganda, I was working at Makerere University in Kampala and the University of Nairobi in Nairobi. Tanzania, I was an agricultural extensionist and working in the Usambara Mountains near Lushoto. So I have a lot of experience working at least in East Africa and traveling through most of Eastern Southern Africa. I haven't been to West Africa, unfortunately. I'd love to go. I certainly listen to a lot of music and hear the food is amazing, spicy. So hopefully one day I'll make it that way. But now I have a family, you know how things go. So, but we'll see. Anyway, let's get started on the presentation. And here we go. Just so you know, this is part of topic 8.3. If you're following with online courses or face-to-face or webinars as part of the EO Africa program, this is just a way for us to organize. So the title of the presentation is use of fPAR, that's the fraction of photosynthetically active radiation, as a proxy for yield estimation and forecasting. The first thing I'd like to do is just give a very quick overview of food security analysis. And the reason I do that is it puts the context of using fPAR and why we use it in crop yield models and what crop yield models are used for. So some of you might already know this. There are 4 sort of official pillars of food security in food security analysis. The first one we address is availability, which deals with the abundance of food, but can also deal with sort of physical access. So if there's a flood or some other physical barrier to delivering food to market. But this is, mostly dealing with the supply side of food security. The next pillar is access, and this is the demand side, more on the demand side. So access can be physical. So basically you can see this here in the lower right-hand side, certain items in the grocery store are missing, and that's because items could not be delivered and therefore not purchased by people who are eating the food. So their purchasing power has been reduced because the food hasn't made it to the market. But it can also be that people are economically poor, and this is just a famous statue in Washington, D.C. from the United States. I'm from the US originally, the US Great Depression, where people had to stand in line for food and drink because they couldn't afford to buy it themselves. And then you have utilization. Utilization deals with the quality of food, the nutritional quality. Say, are you getting enough vitamins? You can see here, this is a plate with the diversity of starches and lentils and rice and many different types of food, which is great. So we say the utilization is high, but it can also be referring to quality in terms of how it's prepared. So you can imagine McDonald's, if you know McDonald's, or some kind of fast food restaurant, the quality of food you get out of there is not very good. It's deep fried, it's processed. So utilization also addresses that issue as well. And then finally, stability, which is really kind of a synthesis of the first 3 pillars. It really talks about how stable your food security is over time. And so you may hear terms like chronic food security or temporary or transient food security. And this is talking about stability. How stable is your food security through time? You can have periods where the food security is high, but then if you're faced with a drought, you lose all your livestock, then of course you're extremely vulnerable now to food insecurity. And so we'd say the system is not very stable. So the reason I give this context is because Earth observation or satellite, what we call satellite remote sensing data, is primarily used to address food availability, specifically food abundance or crop yield. This is where most of our work is done. And food production in kilograms is a product of crop area or cropland, croplands under cultivation. They can also be fallowed temporarily. There's a nice FAO definition of this, basically. So that's measured in hectares, and a lot of work is being done in remote sensing or Earth observation to estimate crop area and crop yield, which is where a lot of the work I do, and this is what the focus of the presentation is, measured in kilograms per hectare. And so you can see here crop yield, the units are kilograms per hectare, production divided by area. And so here's a formal definition. Crop yield is the marketable fraction of above ground biomass accumulated over a growing season for food, feed, fiber, or oil. So another way of looking at it is you have biomass accumulation, the amount of green stuff that crops accumulate over the growing season. Only a fraction of that is used for yield. That's the reproductive efficiency, so the grains that are produced from that biomass. Why would you want to measure crop yield from space? So I'm going to pose this question to you all. Maybe you could write in the Q&A, why would you want to measure crop yield from space? Yeah, it saves time, Eunice. That's great. Yeah, remote sensing offers frequent information consistently at pretty high spatial resolution through time, large area. You can have a footprint of 250 kilometers, 300 kilometers by 300 kilometers, wide coverage, fast. Yeah, what about applications? Can you think of some applications? Scalability, yeah, good point. Yeah, good, nice answers. Ground surveys are expensive and laborious. Yeah, that's right. So pretty typical, but can you think of some applications? Why would you use crop yield? What do we use it for in agronomy, let's say? Forecasting available food. Yeah, of course. This is a very important application. We can now sometimes forecast with reasonable skill the next season, the next year, certainly within the growing season. So if you have some information, say on the reproductive phase of the crops under study, that gives you a good indication of what the final yield will be at the end of the growing season. Yeah, comparability, right? So you can see which regions might have lower or higher productivity and the factors behind that. Yeah, this is something that's very difficult to do with field studies because field studies are isolated. So you might have some understanding of what's going on there in the field you're looking at, but you have no idea how that compares to other fields. Maybe you're not doing so bad, or maybe you are, and that's what remote sensing can help do. Crop mapping, yeah. Yeah, so I think decision making, food supply chain preparedness, sure. Okay, so I think you get the idea. So let's, I provide some generic answers here too, which you can see. Yeah. And so this is just a few of the applications. So I liked the answers. You gave both the kind of logistical reasons why we use remote sensing, but I was looking more for the application, but some of you also answered that way too, which is great. So yeah, these are one of many things that we do with crop yield information, and we'll briefly touch on these as the presentation goes. So you can see forecasting was mentioned, harvest, residue management. So the amount of biomass that isn't used for harvest or yield, that's maybe left on the ground, sometimes tilled back into the soil or burned. This is what we mean by residue management. Crop variety performance, this is a huge topic, because not only do certain crops perform better in certain areas, but different hybrids, GMOs perform better or worse in certain areas. And this can somewhat be assessed with remote sensing. Irrigation, when and where to water, similarly with fertilizer, tillage. Yeah, so you can see a lot of different applications. Yeah. Okay, so the learning objectives of today are just basically, by the end, you should be able to explain what we call the big leaf concept of light use efficiency. So this is the modeling approach we're using. And the reason I start with this is there are more sophisticated models, but these are the models I use. I think they have a lot of advantages for large area applications. And also they're simple. They don't require a lot of data. And for those of you who are modelers in Africa, in many places, we don't have the data we need to run our models. So that's always in the back of my mind when I'm proposing new methods, or does it use free and open access information? That's first priority. And two, does it require a lot of input data? And then we're going to compare the big leaf approach to what we call the radiative transfer or multi-leaf modeling approach. So this is another common approach to estimating yield from light use efficiency, but we'll get into this. It's a bit more complicated and hasn't been widely used over large areas. And then finally, I'll take you a bit step by step through a common big leaf model, just so you understand putting all the pieces together that we talk about, including fPAR, which is the main constraint on life use efficiency. Yeah. So let's start now with just a few basic equations of how crop yield is estimated from essentially biomass accumulation. So you can see the formula here. Crop yield is a function of what we call NPP. This is net primary productivity. So net primary productivity is basically the amount of carbon, which is assimilated as biomass over a growing season minus respiration. And respiration we'll get into in the subsequent slides. Respiration deals with the carbon sort of lost. So you gain some carbon for growth and maintenance of crops, but you also lose some through the metabolic processes. Now, this part is very important, but it doesn't tell us the full story. So you may accumulate a lot of biomass. For those of you who have worked in farms, maybe you've seen, I used to work a lot in cotton, and cotton in particular is very striking. Like it can produce a lot of leafy green, a lot of biomass, but then at the end of the day, it doesn't produce a lot of cotton. Whereas if you constrain the crop, you stress it a bit with water, withdrawing water or something like that, it grows a lot fewer leaves and produces a lot more cotton, right? So that's what I mean. MPP is not necessarily the best predictor or indicator of crop yield. You have to account for these other factors, and I show them here. These are the harvest index, the root to shoot ratio, so how much biomass is above ground compared to how much is below ground, and the moisture content of the grains. So these factors all basically are estimated either from remote sensing or from the literature or measured in the field to basically tell us what fraction of this biomass actually is crop yield. So why do we measure yield with Earth observation? And we talked about this just a moment ago. Yield is a function of three factors: genetics, environment, and management. You may have seen this equation somewhere, GEM. And so basically, there are large spatial and temporal variations in these three factors. Genetics, you're referring to how it may assimilate carbon. We'll get into this. You may have heard of C3 photosynthesis, C4 photosynthesis, but also the different crop types and crop varieties. This all deals with genetics, environment. Now we're talking about climate, let's say soil properties. Management goes a bit further, also deals with, say, fertilizer application, tillage, no-till, residue management, these kind of things. Right? So all of these factors combined give you an estimate of yield. Of course, with remote sensing, we're primarily focused on the environment and genetics, but we can also indirectly, with the approach I talked to you about today, deal with management as well. So yeah, remote sensing, as some of you already pointed out, can account for large spatial and temporal variations in crop yield, which we see. Yep, let's see. I see a hand raised. Did you write in the chat, or do you want to use your microphone and ask a question? Or are you using the chat? No, I don't see. I see a hand raised, but okay. Well, until they're ready, I will just carry on with the presentation. So the main goals of using Earth observation to estimate crop yield are to estimate the size of what we call yield gaps. So this is the difference, and we get into this in the next slide, the difference between the potential yield, what we could get if the environment was working in our favor, the genetics, the management, everything was perfect. This was the maximum yield we can obtain versus the actual yield. So yeah, things aren't perfect. The world isn't perfect. So there are things that happen. And so the attainable yield is actually something less than the potential yield, and that creates a gap. And so then not only estimating these gaps, but determining why these gaps are happening. This is why we use Earth observation to estimate crop yield. It's the main two reasons. Okay, so this is taken from Jones, one of the papers I provided with you, to you. And I find this paper really useful because it goes into, basically breaks down how crop yield models work. So you can see there's defining factors, what we call limiting factors, and reducing factors. So these are the main factors that affect the crop yield. So the first one, the defining factors, these are sort of the environmental conditions and the basic characteristics of the crops that you're using that basically define the potential yield. So this is the maximum yield you could get given the current concentrations of CO2 in the atmosphere, the amount of incoming radiation, the temperatures, and also the physiology, the phenology, the canopy architecture, and these are largely defined by the crop that you're planting. And so this is the most you could get out of this crop or this field or this system. But then, of course, there are other factors, limiting or regulating factors. So these are things like nutrients and water. These are things that the farmers have sort of direct control in regulating, which can affect the productivity of the crop. And so this is where most crop yield models stop. You have the defining factors and the limiting factors, and that's how you estimate yield. But another area are worth including in the models, but it's very difficult because you need a lot of data to do this, are the reducing factors. So these are things that we can say the farmers have less control over, and that's why they're not limiting factors. So things like weeds, pests, disease, pollutants. So the farmer has some control, obviously, if they use Roundup, which kills weeds. It's a kind of defoliant. Pests and disease can be controlled to some extent by pesticides, and pollutants not so much, right? And so you can see, yeah, as I said, the yield gap is the defining or potential minus the limiting plus the reducing factors. So this is a basic overview of most crop yield models that we use. What we call process-based, not data-driven like machine learning. That's a different category of modeling. Yeah, so we don't look at the reducing factors. So how does it all work? Now, just a brief, and I'm sorry, this presentation is meant for a broad audience, so some of you might already know this, but I'm just going to give a quick overview of photosynthesis because this is the engine behind NPP and light use efficiency. So you can see here, this is a schematic of photosynthesis, and essentially what happens in photosynthesis is water is taken up by the roots and desiccated, meaning broken down into its constituents, hydrogen and oxygen. Sunlight is absorbed. Sunlight is basically the source of energy, and carbon dioxide is taken up in leaves and stems through what are called stomata. These are like pores in your skin. The plants take up the CO2, which is broken down during photolysis, again, into its constituent elements. So you have oxygen and carbon, and that carbon, oxygen, and hydrogen is recombined to form what we call sugars, specifically glucose. And these sugars provide energy to the plant for growth and maintenance. And so you can see here, this is a schematic of the Calvin cycle and the desiccation of water here on the left. So this is sort of the engine behind photosynthesis. And this occurs in what are called the chloroplasts. And this is what remote sensing spends a lot of time doing, is detecting the response of chloroplasts to light, because chloroplasts love red light, particularly during the growing season. They use red light during photosynthesis, and they hate green light. That's why they're green. That's why plants are green. And also because of the plant structure, they reflect or scatter light in the near infrared. Yeah, I see there's a question. Afternoon, please, would you send us the recording is important. Okay, so I pasted the link in the Q&A. If you want a recording of this particular presentation I'm giving now, no problem. I can do that. I'll talk to Yob, he's our technician. But if you just want a clean recording, there is one on Vimeo already. Okay, so let's carry on. Okay, so once the sugars are formed, they're then broken down into something called cellular respiration. So this process breaks down the sugar to create energy. We call these energy packets or batteries ATP. And these ATP are used by the plant to grow, to maintain functions, this sort of thing. And so respiration happens in every living cell in plants, and it happens in 3 stages. And right now, I just want to distinguish between what we call autotrophic and heterotrophic respiration, because it's important here. Autotrophic respiration is the respiration that plants use. It's respiration that happens by light harvesting plants. It can happen day or night, but it's light harvesting plants that use it. Heterotrophic respiration, on the other hand, is used by little critters like nematodes and worms in the soil who don't use light. And we're essentially heterotrophs as well, right? We get our sources of energy from something other than the sun. We eat food, right? And the reason why it's important to make that distinction is because the first process of respiration, glycolysis, breaks down this sugar, this glucose, into pyruvic acid. This is only occurring in heterotrophic respiration. Autotrophic respiration has 2 additional cycles. One's the Krebs cycle and one is electron transport. And basically, these 3 processes just strip every possible energy they can from the sugar. And the byproducts are carbon dioxide and water. And so you can imagine autotrophic respiration is more efficient, right? It produces 3 energy packets or ATPs versus 1 in heterotrophic respiration. Okay, so just to recap, here are the main differences between photosynthesis and respiration. So in photosynthesis, sugar molecules store energy, right? So photosynthesis, they make glucose from CO2 and H2O, and this sugar stores energy, and then during respiration, that energy in the sugar is released. Much like when we eat sugar, right? We release energy as well. It's just we're getting our sugar from some food source, whereas plants make it themselves from the sun, water, and CO2, which is pretty remarkable. So photosynthesis, CO2 and H2O are absorbed, CO2 and H2 are released during respiration. So that's what I meant earlier when I said respiration is a cost. So you have CO2 accumulated by the plant, but also which becomes biomass, but some of it is lost to respiration. So photosynthesis increases biomass, respiration uses biomass. Photosynthesis is a light reaction. Respiration can happen light or dark, but and then finally, it occurs in the chloroplast, photosynthesis, respiration in all living cells, and then oxygen is a byproduct, and in respiration, it uses oxygen for autotrophic respiration. Are there any questions so far on this? This is a very quick overview. Of photosynthesis, because it's important to understand where the terms like NPP come from, which is your biomass accumulation. Oops. All right. Okay, so now that we have an idea of what photosynthesis is, now I can discuss another important concept, which is called gross primary production, or GPP. And this is essentially what these remote sensing crop yield models are estimating. They're estimating GPP, which is basically the biomass accumulated over 30 minutes or hourly or daily, depending, or monthly, depending on the data that you have. And so this GPP is the biomass accumulated over a day or hour or whatever, and then it's summed over the growing season, defined by the start of season and end of season. And usually respiration is removed, or the cost is removed at the end of the growing season to get at NPP. So this is respiration due to growth and maintenance. And there are different ways of estimating the respiration from remote sensing, or sometimes it's assumed respiration is a constant fraction of GPP. And there's a lot of evidence to show that respiration on average is about 50% or a little less, maybe 40% of GPP. And this is a fairly conservative estimate, but there are other studies that use, say, NDVI, the Normalized Difference Vegetation Index, to estimate respiration as a function of GPP. And then I show here the two, some older studies even use ambient air temperature to derive respiration from GPP. And so the units of GPP as NPP and crop yield are kilograms per unit area, can be grams as well, or tons. You don't have to use meters squared, hectares. There are many different flavors. Okay. All right. So now let's break this down further. And now the next slides are really trying to align important aspects of photosynthesis with different components in a crop yield model. So the first thing these models consider usually is what we call quantum efficiency, or CO2 efficiency. Basically, how good is the plant at assimilating CO2, turning CO2 into sugar, right? And that's, there are a few different ways to do it. Some use what is called the fPAR model of photosynthesis. The model I use is Collatz, 1991 and 92. Some just assume there are no differences, or there's lookup tables, or these different things. We don't need to get into the details, but the main features we're looking at are what are called C3 plants and C4 plants. C3 plants, most crops are C3. I show some examples here: rice, cassava, wheat. C4, the main C4 crop is corn or maize. And basically what you need to know is C3 plants follow the Calvin cycle, as I said before. And the Calvin cycle involves the Rubisco enzyme. This is the enzyme that sort of catalyzes the reaction of basically forming sugars, right? Which are then broken down during respiration. The differences is C3, C4 plants have an additional step. This is the mesophyll. This is the sort of middle leaf layer. They have an additional enzyme called PEP. And what this PEP does is it concentrates the CO2 in the mesophyll, this layer in the leaf. And because it concentrates the CO2, that means more CO2 is available for the Calvin cycle. So C4 plants are more efficient at using carbon. And you can imagine these plants do particularly well in dry climates where they can't lose a lot of moisture through their stomata via the process of evapotranspiration. So this is a nice map, Powell 2012, produced by a colleague of mine, basically where they use climate data to sort of map where C3 plants tend to grow and where C4 plants tend to grow in South America. I think eventually they want to make these maps global, but for now, I think they've only published these maps for South America. So if you can see the percentage of C4 plants in the blue here is quite low where there are forests. So trees are C3, I think almost exclusively. I'm unaware of any C4 trees because C4 photosynthesis evolved much later with grasses. And then you can see here where there are higher percentages of C4 in the east of Brazil. These are dominated by savanna, grasslands, maybe some shrubs, mixed systems, but involving grasses or crops, this sort of thing. Yeah. And so these climates are drier and hotter as opposed to the Amazon, which is really apparent here in blue, which is humid. warm but humid. So there is one other photosynthesis process. We don't consider it. It's called CAM. So these CAM photosynthesis these plants are even better adapted for dry climates, but there aren't many CAM crops. The only one I know of is pineapple. Okay, so now this is one component, the CO2. The quantum efficiency, this is one component of crop yield models. The next one deals with radiation. And so here I just highlight sort of the three main aspects that these models address, either directly or indirectly. The first thing is the quantity of light. So this is your photosynthetically active radiation, or PAR. This largely depends on seasons. So obviously in the spring and summer, light, particularly at northern or southernmost latitudes, is more abundant. So there's more light. Also the leaf angle and the solar angle is also very important, because this helps us to address direct versus diffuse light. So many of you might think direct sunlight is the best for plants. They love it the most because the light is shining right on the plant. But what ends up happening actually is the light only exposes a certain part of the crop, not the whole crop. So greenhouses, one of the reasons they're so successful is because they diffuse light. So you can see that illustrated here. These solar glass panels are diffusing light. So instead of light coming from one direction and exposing just one part of these tomato plants, light is bouncing all over the place. And so the light gets under the leaves, the stems, you name it, right? So diffuse light is actually better for plant growth than direct light. Quality of light is also very important. And I give an illustration here of a greenhouse. I live in the Netherlands. As you may know, the weather in the Netherlands is not that great. It's pretty cold and cloudy most of the time. So they grow a lot of food in greenhouses, and they basically change the part of the spectrum throughout the growing season, which they use to grow, because plants preferentially absorb different times of the year, different parts of the spectrum depending on their position in the growth, their growth stage. So this plant here, for example, when it was sprouting, probably was exposed to UV and visible blue light, because this is what stimulates growth. And then later in the growing season, particularly around the flowering stage, the reproductive phase, the light probably switched to red. And red light is what plants, because of the chlorophyll and photosynthesis, love the most. And then another factor which is important but not easily captured with crop yield models is what we call the photoperiod. So photoperiod basically deals with the amount of light when, and certain plants have certain photoperiods than others. And so there are sort of two main differences, and I show that here on the right. You have these what are called short day, long day crops, and long day, short day crops. And so what a short day crop is here on the left is, you can see during the early part of growth, the days are long and the nights are short, but during the reproductive phase, the nights are long and the days are short. So it actually needs longer, cooler nights to reproduce. And so a good example of this is winter wheat. Winter wheat is planted in October and usually harvested in the spring, maybe a bit later. And so the days are rather short when it starts reproducing. And so this is an example of a short day. Of course, the other one, which is what we're probably more familiar with, are these long day crops. So like maize or several other different types of crops where the reproduction happens when the days are long and the nights are short. So this is maybe grown in the summertime. Yeah. And then of course you have day neutral, which these plants are, they don't care really whether the days are long or short. And so you can imagine if you try to plant winter wheat, say in the summertime, or corn in the fall, you're going to get crop failure because the photoperiods aren't properly aligned. Okay, so these are the main components of crop yield models. Now, what do we do with this information? And there's two different approaches. So the way we're mostly going to talk about today is the big leaf approach. And the big leaf approach uses this term, which we call fPAR, fraction of photosynthetically active radiation. There is another approach, which you may hear about, which is called a multi-leaf approach or a radiative transfer model approach. So the big difference is big leaf approach assumes that the canopy is homogenous. And you can see that here. Basically, it's a flat sort of surface, like a pixel. And when light hits that surface, there's some reaction with the plant. And so we're only really seeing what's happening at the top of the canopy as detected with the pixel. And the reason this works is because there's a tendency for light within the canopy to sort of normalize. And so a good estimate of the top of that canopy tells you also what's happening below the canopy. But of course, if the canopy is very heterogeneous, there's a lot of leaves, they're oriented different ways, there's a lot of structure, then this doesn't work. This big leaf approach doesn't work so well. But of course, by assuming this, you require a lot less data and information. And so for large area applications, this works pretty well. So in a paper I published, for example, I estimated crop yield pretty well across the United States at 250-meter resolution with MODIS data. Right? That would not have been possible with a multi-leaf approach. Okay, so now the nuts and bolts of fPAR, which is basically the most important constraint on big leaf approaches to light use efficiency. And I illustrate how it works here with what we call a septometer. So some of you who have worked in the field maybe have used this instrument. What a septometer does is it has a sensor at the top or above the canopy, which collects incoming solar radiation. And then this wand here you put under the canopy, and it measures the light that basically passes or is transmitted through the canopy. And the difference between the light received at the bottom of the canopy and the top of the canopy is essentially your fraction of photosynthetically active radiation, your fPAR. So basically it's assuming everything in the canopy is absorbing light, and that's your fraction. And it works on the principles of Beer-Lambert law. The transmitted PAR equals the PAR incident. This is the PAR or the light received by the sensor at the top of the canopy as a function, exponential function of this extinction coefficient K and Z, which is in our case, the path length or leaf area index. So the leaf area index is essentially a two-dimensional measure of how much leaves, the area of the leaves in the canopy. And you can see this is illustrated here. There's a nice relationship between, exponential relationship between leaf area index and PAR, fPAR as a function of the extinction coefficient. So basically as leaf area index increases, that means there's more stuff in the canopy. The fraction of photosynthetically active radiation is going up because more stuff, it absorbs more light. Pretty, hopefully straightforward process, but this is super important for understanding light use controls on crop biomass accumulation and yield. Are there any questions on this so far? This is a very important concept to understand in these models. Okay, I don't see any questions so far, so we'll continue. So just to let you know kind of how these operate, it's just for your information because many of you might decide not to use the big leaf approach, and there's no problem doing that. There's also many people, a good colleague of mine, Christian Vandertal at ITC, developed what's called the SCOPE model, S-C-O-P-E, SCOPE. And this is a multi-leaf or radiative transfer model. So it's much more sophisticated, as you can see here on the right. It consists of two components, a leaf model and a canopy model. And basically, the leaf model takes into account the biochemical processes involved in photosynthesis. So chlorophyll absorbing light to make sugars, basically, these kind of processes. It also accounts for accessory pigments, which I get into briefly in a subsequent slide, leaf water, these kind of factors. And then these leaves are essentially scaled over several leaves, and then eventually with the canopy model over the entire canopy. And so the canopy part deals with structure. The leaf part deals with biochemical, the chemical side of photosynthesis and light use efficiency. And so the canopy model takes into account canopy structure, like leaf area index, and also the sun sensor geometry, the orientation of the sun with respect to the canopy. And then you can see here very sophisticated three-dimensional models put these reactions scaled to the canopy. They used to be like a kind of slab model. You can see this here. Now they're scaled over cubes, and these cubes are then scaled over, say, an atmospheric boundary layer. And so you can account for different vegetation types, soil, water, built up structures, this sort of thing. So very sophisticated, very good models. But of course, if you've ever heard the term of parsimony or propagation of error, when you have a lot of inputs and you don't know them very well, because over large areas you can't, you have to make assumptions. The question arises, is it better to have a model with a few parameters that don't require a lot of assumptions, but you know those parameters really well? Or is it better to have a model that's really good, but it has a lot of assumptions and you don't know the inputs very well, and so you kind of make some guesses? And so that's something, you have to think about if you use this approach. So this is a question for all of you. So if you can write in the Q&A, sorry. Which crop has the lowest fPAR on average? The fraction of photosynthetically active radiation. Cotton, rice, or maize? So if you can, please write in the chat what you think your answer is. Sorry, not the chat, the Q&A. Okay, a few of you have written already, a couple of you, cotton, some say rice, a lot of rice. Can anybody tell me why they wrote cotton? Or you can also say, if you want. Why does cotton have the lowest fraction of photosynthetically active radiation, fPAR? Anybody willing to give an answer? Alternatively, can somebody give an answer why they wrote rice? Leaf angle. Anonymous attendee. No name given. Okay, anonymous attendee. That's you're on the right track. So let's just somebody else. Lesser leaf area. Well, that's hard to tell. That could be, but if you've looked at rice potentially has a lot of leaf area. Because there are several shards or several leaves, but it's actually more to do with orientation. So the anonymous attendee who said leaf angle, I think that's closer to the correct answer. But you're thinking in the right direction at least. So indeed, the right answer is rice. And so I include some diagrams here so you can get a visual. So cotton has a very high fraction of photosynthetically active radiation because the leaves are what we call planophile or horizontally oriented, and the leaves are very broad. right? So if you can imagine, if light's coming through the canopy, a lot of that light is intercepted and captured by the canopy. And so if you have your septometer below, you're not receiving a lot of light. Similarly with maize also, if you've worked in maize fields, they're pretty dark. The leaves are more erectophile, more vertically oriented than say cotton, but still pretty much a planophile, large, leafy, sort of horizontally oriented leaves. Whereas rice is what we call strictly an erectophile, right? So the leaves are very vertically oriented. So a lot of the light passes through the canopy and is not intercepted by the rice, right? So on average, over growing season, it probably has a lower fPAR than the others. Yeah, somebody raised their hand. Yeah, it may also, good point, Edward. Of course, yeah. So this was just a question to get you thinking. Of course, a lot of factors are involved. So yeah, if cotton, for example, was very spread out and your target was a field or over a larger area, your scale of observation, we call it, then of course the fPAR could be lower because it's scattered. Whereas rice, for those of you who have worked in rice, it's planted pretty close to one another. But I was more specifically addressing like one particular plant. But you're absolutely right. Good point, Edward. Thank you. Are there any questions, other questions so far? Let's see here. Why reflected radiation? Somebody asked why reflected radiation is not taken into account with fPAR. Well, fPAR is dealing with the light that's absorbed by the canopy. So light reflected off the surface of the canopy is not used during photosynthesis. But if you're referring to light reflected off the soil to underneath the canopy, fPAR can account for that, like in a multi-leaf approach. But with the approach I use, the big leaf approach, no, it can't. But if you're talking about light reflected from the soil back into the canopy, absolutely, this can be taken into account. And the septometer that I showed also, it's not a perfect instrument. This is used to get a rapid estimates, pretty high accuracy. And so it only accounts for the light that passes through the canopy, but not necessarily hitting the soil and reflecting back. But I like the septometer because I think it creates a nice visual between the big leaf approach and light use efficiency. Can we measure the fPAR by solar-induced fluorescence data? That's a very good question, Alexander. Fluorescence, SIF for short, is an emerging topic. There's a great paper by Guan, G-U-A-N. He's a professor, I think, at the University of Illinois, Urbana-Champaign. And he wrote a really nice paper where they compare SIF to microwave and hyperspectral remote sensing, I believe, for crop yield estimation. So SIF, basically, for those of you who don't know, there's a very narrow region, I think around 750 nanometers in the what we call the near infrared. This is light that's actually energy that's emitted by the plants during photosynthesis. It's a very small amount. It's like 1-2% of the total energy that you could measure, say, with a sensor. So it's very small, and this is one of the challenges, but it's very tightly coupled to photosynthesis and production. So in theory, maybe not fPAR per se, but you could certainly get at GPP and NPP with SIF. But there's some challenges. And like I said, one of it, the amount of energy is small, and the current satellite that's using it, I think it's called GOME, G-O-M-E, is very coarse resolution spatially. There are new missions coming up, ESA Flex, F-L-E-X, for example. This will be launched, I think, 2025. And so this will measure SIF at pretty high resolution and like frequently over the globe, right? So like a Landsat or Sentinel-like mission for SIF. So I would certainly look out for that. I think that's going to be a really hot area of research in the coming years. But that's a good question, Alexander. I hope I answered it. And let's see, I'm just scrolling through the chat now to see if there's more. I think I answered most of them. Yes, we will, Dr. Subhash, covering fPAR. Yes, as long as you stop asking questions. No, I'm joking. Yeah, so yeah, SIF, also the temporal resolution is not very good, but the spatial, I think, more so, because you can imagine if you're trying to trace a signature of that's only 1 to 2% of reflection / emissions from the ground. If you're looking over, I don't remember the resolution of GOM, is it 10 kilometer by 10 kilometer or something like that? Or even more, maybe it's 50 by 50. I think it's 50 by 50. Yeah, good luck. But there are missions coming up. So please be on the lookout for those. Okay, so let's move forward. So now we're going to talk about vegetation indices, because these are the main ways that fPAR is calculated. Normalized difference vegetation index is the most common. This paper by Tucker is a great start. He was not really the first to publish, but he's the most famous. He's the one that's recognized. But in fact, there were a few publications before this. And basically what it relies on is the fact that healthy plants scatter strongly in what's called the near infrared. So if you look at this image here, this is a natural visible image of trees and grasses and mountains and a house. This is what we would see in the near infrared, if we could see in the near infrared. Our eyes do not detect near infrared. And you can see here that indeed, even more so than green light, you would see very bright features from the grasses and the trees because they scatter light. And that's at the leaf level, that's because near infrared is not liked by chlorophyll. It doesn't preferentially absorb near infrared. At the canopy, or it actually, and there's scatter in the mesophyll, the inner leaf layer that I referred to earlier. At the canopy level, it's more to do with the orientation of the leaves in the canopy. So there's more scatter there as the canopy grows and becomes more structured. And then it also relies on the red light. And this red light, as I said earlier, chlorophyll preferentially absorbs red light. Less so blue light, but more early in the growing season. Red light, though, throughout the growing season is a good indicator of photosynthesis and production, right? So it combines this in a sort of normalized ratio, and that's to account for some sources of error in the individual bands, the channels. So if you look at the channels themselves alone, they're susceptible to topographic effects, soil moisture effects. They're also affected by atmospheric constituents, the solar angle, these sorts of things. So NDVI sort of corrects for that. Now, NDVI is the most famous index. It's still widely used to estimate fPAR. But I also recommend, if you click on this slide, this will take you to a website. This is Harris. They develop NVIDL. It's a GIS software. Maybe some of you are familiar with. You have to pay for it, unfortunately. But the website is free. And basically, it shows you different indices based on what you're trying to estimate. So if you're looking at, say, broadband greenness or narrowband greenness, light use efficiency. So if you click on one, then you get all the different indices that are available. And then let's say EVI, this is a very famous one. You can click on that, and then it gives you the equations and the source of the index where it was hopefully first published. So this is, I think, a nice resource. It doesn't give you all of the indices. There are so many indices out there. Somebody said for every remote sensing scientist, there's an index. But it's a good place to start, I would say. Okay, so are there any questions? All right. So also, Dr. Subhash, we are covering the computation of fPAR and crop yield, but I should add, if you're actually looking to do a practical or something like that, that's not the point of the webinar. The webinar is to just introduce the information to you. There will be later in the year an online course where we take this information and actually go through computations through practical assignments using QGIS and also the EO Africa Innovation Lab. So if you're looking for more hands-on stuff, that's not the purpose of this webinar. I should have explained that maybe in the beginning. But if in fact you're just looking for sort of a brief introduction, this is the place for you. Okay, so now we've talked about fPAR. fPAR is usually estimated from vegetation indices, but there are other factors we have to consider that are constraints or modify how well plants photosynthesize, that NPP, right? So another, and there are different models. Some use some constraints, some use others. So I'm going to try to cover them as much as possible. Is there any technical exercise, Alexander? There is a technical exercise that is part of this webinar, but that's not the point of the webinar. Sorry, it's a presentation. If you want a technical exercise, I can provide one for you. Do you want that, Alexander? Alexander, are you still there? Okay. Yes. Okay, Alexander. Thanks. You took a while. I hope you're not sleeping. Okay. Now, great. So I will try. I don't know how to do that easily with Zoom. I will try to paste it, the PDF, and then you can go through it if you want. Okay. Great. So let's continue with the webinar. Yeah, so webinars are mostly for presentation. There's reading assignments which could help you understand the material more, but the idea is then if you want to learn more and do more hands-on stuff, you should do the online courses available through EO Africa, or if you're lucky enough to go to one of the face-to-face courses. We will also be doing a face-to-face course later this year. Where, I don't know yet. I'm pushing for Zanzibar because I miss the beaches, but I don't think I'll be so lucky. They'll probably have it somewhere else. Okay, so leaf age. This is another constraint on photosynthesis. So you can imagine during the reproductive phase, photosynthesis slows down and the leaves begin to lose their chlorophyll. And because of that, yeah, they're not absorbing as much light. And you can see here, different vegetation indices can assess this. This is just a diagram here on the left for NDVI. So NDVI, when it's very high, that tends to mean the leaves are green, photosynthesizing, happy, producing biomass. So a value of 0.8, even 0.9 is feasible. But as the leaves age, they brown and eventually die during the brown down or senescence phase. The leaf area index goes down. And so the browner it gets, the lower it gets, say below 0.3, 0.2 NDVI, it's basically the canopy is starting to look like the soil from a reflection point of view, from an NDVI point of view. They kind of look the same. So what this function here on the right does in crop yield models is it accounts for this change due to greening up and browning down. And so what it's showing here is the change in GPP, the biomass accumulation versus the day of year. And so this is just an illustration of a common NDVI profile, right? Sort of green up phase, brown down phase. The peak NDVI here corresponds to the reproductive phase when there's organs for reproduction being formed, and eventually grains will fill, and that's your yield. And so basically during the green up phase, this function has no impact on NDVI. So basically NDVI is telling you the plant is producing, it's happy, it's photosynthesizing. But as the NDVI goes down during the brown down phase, now this leaf age is kicking in. So not only is the NDVI weighting the function, but now leaf age, it's weighting it even more. And this is just a formulistic way of sort of accounting for this browning effect in the NDVI. We see it in the NDVI signal, but it's also, this function helps reinforce that sort of relationship in the model. Yeah. Okay. Good question. So I'm also trying to answer questions as we go. Edward, if I didn't say already, at which stage of crop growth season is NDVI critical? So NDVI tends to be maximum around the reproductive phase. So if you know crop phenology, after the reproductive phase, basically energy is already starting to be diverted to forming the reproductive organs and to grain filling. And so photosynthesis tends to decline as the season progresses after the reproductive phase. Everything before that is all the energy is being put into making those organs, right? And so, yeah, NDVI, it's not perfect, but usually around maximum NDVI is when the reproductive phase happens. And in fact, a lot of models, there's a great paper by Johnson, I think it's 2016, and basically he uses the reproductive phase to predict end of season yield. So it's what we call within season forecast of yield. And that's because, yeah, if you have a good idea of what's going on around reproduction, that affects the remaining part of the season. So if you do that practical that I'm going to try to share with you somehow, that's in fact what you discover in the analysis. You use Sentinel-2 data to identify an index and what time of the season is good at predicting yield. Okay, great. Alexander gave his email address. So yeah, that works. Okay, yep. As you know, NDVI is about greenness and it cannot account for GPP and NPP. Is it possible to use EVI? Yeah, Alexander, that's a good point. And look, there are different indices. They all have their pros and cons. NDVI is good at measuring greenness and also abundance. And actually, it forms the basis of most light use efficiency models to date. It's what we use. It's not perfect. It has a problem with soil background. It's sensitive to soil wetness. It does tend to saturate, what we call saturate, at very high values. It's not good at resolving, say, leaf area index at very high values, so very dense canopies. So it has problems, but it's still used, and it still does very well. Now, that's not to say you shouldn't look at other indices. So the Enhanced Vegetation Index, EVI by Huete, 2002. This was first published. Huete is Huete. Huete has some advantages. It tends to be better at measuring leaf area index. It doesn't have the saturation problem that NDVI does. So yeah, I didn't have the time to go over all the indices, and I'm sorry for that. I focused on NDVI because this is what most of the models use. But certainly, yeah, there are other indices out there. And EVI, under a lot of circumstances, is probably better than NDVI for fPAR. Yep. Cool. NDVI. Yeah, again, Alexander, guys, look, I would suggest moving forward in remote sensing, you're not so, what's the word, religious, or I don't know, so strict about what indices to use for what. I always approach a problem by saying, what do I have and what do I need? And I'm open to trying different things. So NDVI solves a number of issues that these other indices can't. So yeah, NDVI doesn't detect pigments, right? There are other indices that are better at detecting, say, green and the accessory pigments, the xanthophylls, the anthocyanins, the carotenoids. There are other indices out there, but they're not widely used. And you have to ask yourself why they're not widely used. And I think one of the issues is NDVI does a pretty good job. Even with all of its problems, it still does a pretty good job. And it's an index that's available over decades, which a lot of these indices you can't. So if you're looking to do like very large scale study in space and time, NDVI is a good way to go, right? So I'm not trying to say that NDVI is the best, but it has some advantages over these other indices. You're welcome to explore them. And of course, we have other, again, that wasn't really the point of this webinar. There are other education resources available through EO Africa on vegetation indices. I have a presentation on that where we compare and contrast these indices a bit more. The main point of this webinar was just to show you the nuts and bolts of the crop yield models, light use efficiency models, and how they're parameterized. And they're typically parameterized with NDVI. Another way to look at it is if you're a researcher or if you're a student and you're looking for new topics, then of course these models are not perfect. And in fact, remote sensing is used in a pretty limited way. And so you might think of some ways of improving the models with these new indices. Or I have a student right now, for example, who's using different vegetation indices from Sentinel-3 and MODIS to detect heat stress controls on production, which isn't widely used. They usually use temperature data. Right? So, but good points, all of you. I'm glad you're keeping motivated. Okay, so again, going through the main constraints that make up these crop field models. The next common one is the temperature constraint. And the temperature constraint usually consists of two components, what we call a cold extreme constraint and a warm constraint, extreme constraint. A lot of models just have the warm extreme constraint. So you have temperature involved in the carbon part, right? As temperatures increase with visco, that enzyme tends to be more active and produce more carbon and biomass accumulation. But there's also this kind of external environmental constraint on photosynthesis, and that's what this FT is. And so you can see here illustrated, this is the percent GPP or biomass accumulation versus the temperature. So in the warm extreme constraint, as temperatures increase, photosynthesis increases and GPP is accumulated until some optimal temperature is reached. And that optimal temperature, if you're lucky, corresponds to the reproductive phase. It doesn't always happen. So there's oftentimes a difference between what we call the optimal temperature, the temperature at which the plants are the most happy photosynthesizing, and the maximum NDVI or other vegetation index of your choice. And then what happens usually as temperatures exceed this optimal is that productivity goes down. And as temperatures become more and more extreme, think of, say, a heat wave or something, the temperatures drop off even more. So other factors that go into this are crop quality. The photo period, as I said, is very limited in these models, but it's sometimes accounted for. Dormancy and hardiness also are important factors, but often not accounted for in these types of models. And so what do we mean by dormancy? Well, like winter wheat, for example, has a dormancy period where it's sort of low activity during the winter time until the early spring, and that's when it sort of bolts. And then hardiness, of course, oftentimes you need root vegetables, wheat as well, needs like a nice cold spell, freezing to stimulate production. Yeah. Okay, so one of the questions, Abdullahi, is it possible to use LAI to detect cotton photosynthesis in Africa context? And the answer is yes. There are different ways to do it. You could use a light use efficiency approach like we're talking about. Leaf area index is the input to fPAR. And leaf area index can be measured from field data. It can be estimated from vegetation indices like NVIDL or EVI or Savvy, or there are a number of indices out there, or you can use existing products. So leaf area index in particular, these are derived products. Modis has a leaf area index product. Also Sentinel-3 through the Copernicus website has a leaf area index product. And so you would put this into a light use efficiency model with other factors, climate factors, say, to estimate photosynthesis and production. You can also do a data-driven approach. I don't think you'll have much luck. Maybe you will. It's hard to say. But with cotton, if it's irrigated especially, you're not going to probably find a good relationship between leaf area index and cotton. So if you production, so if you use a data-driven approach, you would need other data to go into the model. Okay, but all nice comments, good questions. I'm glad to see the interest. Okay. All right, so we talked about carbon so far, carbon use efficiency. We've talked about PAR, the amount of light, quality of light. We've talked about the fraction of the canopy that absorbs that light. We've talked about leaf age. We've talked about temperature, and now we're talking about moisture. So these are all constraints, as I said, one by one we address, which form the nuts and bolts of a light use efficiency approach to crop yield modeling. And this is the main way remote sensing data is used to estimate crop yield. Of course, there are data-driven approaches. I don't go into those because they're not as, to be honest, not as interesting. They don't really tell you why plants are doing what they're doing. But of course, they can yield to higher accuracies if you have good data and you know what you're doing with machine learning. If you carefully select model inputs, etc. So Dr. Subash, are the cold temperature curve in area specific? No. So one thing it's a good point. I should have maybe mentioned is the light use efficiency approach is not area specific. It's not even crop specific a lot of times. Like I said, these are used for large area applications for monitoring. So if you look at, say, the Copernicus product, NPP, or they call it dry matter productivity, DMP, this is a light use efficiency approach, or WAPOR, maybe you're familiar with. WAPOR estimates crop water productivity across Africa. It also uses this big leaf approach. And so for large area estimation, like I said, you have to make certain assumptions. Now, is this approach better than, say, DSAT or ERISA, these field level kind of crop yield models? The answer is no. At the field level, but over large areas, probably yes, because ERISA and DSAT, APSUM, these crop yield models don't work well over large areas because they're extremely complicated and they have a lot of data requirements and this sort of thing. Yeah, but good question. Okay, so now water and humidity. The main control is water vapor pressure deficit, VPD, or relative humidity. These are the main inputs to estimate the constraint of moisture. Sometimes soil moisture is used if that data is available. This is becoming more common now with the emergence of Sentinel-1 and SMAP and other microwave-based products. So, and I think these are better at estimating the control of moisture on the canopy, but sort of the older models or traditional models use vapor pressure. And this is based on the principles of Boucher's hypothesis, B-O-U-C-H-E-T, Boucher. who basically said, hey, if the atmosphere above the crop is moist, then the crop is probably moist and not stressed. If the air is very dry, then the crop is probably pretty stressed from a moisture point of view. And this is illustrated here on the right. You can see this is, say, a leaf layer, this dotted green line that separates the atmosphere from the leaf. And the reason this happens is if you have these water molecules here in the atmosphere, if there's very few, then water tends to be drawn out of the leaf through evapotranspiration. And that's because if you know sort of basic plant physiology, things move from high pressure to low pressure. So if you have a lot of water in the leaf, it tends to want to go out to equalize with the surrounding air. However, if there's a lot of moisture in the air, it's very humid air, then the deficit is small, and so you don't see so much movement of water across the leaf boundary. Right? And so you can see this illustrated here in the lower right-hand side, GPP, biomass accumulation versus vapor pressure deficit. So as you can see, for very sort of low vapor pressure deficit, meaning very moist air, say between 0 and 1 kPa, pascals, there's not much change in productivity. But as you increase that vapor pressure deficit, more water is being lost to the atmosphere. And so the GPP goes down. Basically, the plants are closing their stomata, not taking in as much CO2 because they don't want to lose the water. And this is, again, why C4 does better at sort of drier climates, because basically the stomata don't have to open as much, so they don't lose as much water because they're able to concentrate the CO2. You can see water is involved in a lot of functions that aren't really directly captured by this approach. Photolysis, the desiccation of water, breaking down the water absorbed by the roots to make sugars. Water helps to maintain turgor pressure. The structure, it's also an important solvent or medium for nutrients and transport throughout the plant, through the xylem. Transpiration, so transpiration has a couple of functions. One of them is to help maintain this sort of turgor pressure, but also it helps keep the plants from heating up too much. So if you have ever experienced on a hot day, if you're outside, you start sweating and that water evaporates from your skin and it cools you down. And transpiration basically does the same thing for plants. And it also helps with root elongation. Yeah, so Dr. Subash, great. Another question from you. And then I would say we have to, if we're going to finish on time, maybe we reserve the questions a bit later. Excuse me. It's great. The questions are great, but if we're going to finish by 4, I think we have to sort of move on, but I will answer for you. The big leaf approach, the big leaf approach, like I said, doesn't require crop-specific information. Basically, it assumes these kind of biophysical constraints are dependent on the data itself and not the physiology of the crop. That said, once you calculate the harvest index, usually the harvest index is crop-specific or area-specific. And unfortunately, at this point, there aren't automated ways. I'm actually looking at methods to automate the process on a pixel basis. But right now, when we run these models over an area, we assume the harvest index for rice is this value, and the harvest index for corn is this value. So that's when the crop type mapping becomes an important component. Okay, great. I hope that answers your question. Now, another factor, again, that's super important, but a lot of models don't handle it directly, are nutrients. And so I give a brief illustration here by Janssen, 1990. This is a very common way of accounting for nutrient constraints on crop production. It addresses the three main macronutrients, which are nitrogen, phosphorus, and potassium, NPK. You've probably seen or heard the term NPK fertilizers. So most fertilizers have abundant supplies of nitrogen, phosphorus, potassium, or might have one or the other in abundance compared to the others, and that's because it's the sort of limiting factor. And so how these models handle nutrient limitation is basically they relate the potential of nutrient supply. You can see that illustrated here on the right-hand side on the y-axis versus, say, a common soil component, pH, soil acidity. This is something that's very easy to measure in the soil. The data is quite common, even gridded data over large areas. Organic carbon, Olsen P, exchangeable potassium. These are some other ones that are used. I highlight organic nitrogen and total phosphorus in red. These, of course, would be the best, but these are the ones that are least likely to be measured in the field or available in geospatial information. And so you come up with this relationship between the three factors and the soil properties. So you can see here, potassium, for example, as pH goes from low to high, it tends to increase the amount of potassium, but then as pH gets very high, the K goes down, whereas phosphorus and nitrogen sort of have opposite relationships. So at low pH, nitrogen is high, and P is high, the phosphorus, and opposite is for high pH. And once you have this relationship, then there's usually an additional step of accounting for the limiting factor, because these relationships would hold only if one nutrient wasn't limiting the other. And that's not the case. Usually one is limiting the other two, right? And so in this case, we illustrate yield versus P uptake. And so in this illustration, you can see an example of P limitation. So P uptake is quite high with the variation in yield, and that's because there's not a lot of phosphorus in the soil. And so the plant's trying to take up as much as it can. Whereas if there's a lot of P, you can see the response between P uptake and yield is quite shallow. The slope is lower, and that's because it doesn't need so much phosphorus. So combining these information on phosphorus or potassium or nitrogen limitation and the relationships with soil properties gets at basically the nutrient availability and then ultimately the constraint on yield. Diving a bit deeper, we've talked mostly about NDVI and other sort of multispectral, what we call multispectral broadband techniques. Multispectral data, basically the spectral information is available over wide bands, say from 700 to 800 nanometers. And so a lot of spectral information is lost. The spectral resolution is coarse. And because of that, you can't get a lot of detail in the information in terms of crop growth and development. So an active area of research, one that I'm involved in actually, is to look at what we call hyperspectral narrowbands, hyperspectral information to predict yield. And so I provided this diagram from a colleague of mine, Mariana Belgiu. This is a paper we published a couple of years ago that illustrates the value of hyperspectral narrowbands. So this is reflectance, which you might detect at the top of a canopy. In this case, it's by the Prisma satellite. It's a new satellite launched by the Italian Space Agency. And it shows you variations in spectra from 400 to 2,500 nanometers. And it does it for three main phases of wheat development: the vegetated here in the green, reproductive in the orange, and in the purple, maturity. And so you can see some really interesting differences. So in the reproductive and vegetation phase, you can see a lot of absorption in the visible range from about 400 to 650 nanometers. And a green peak around 550 nanometers. You can also see absorption around 690 nanometers, and that's because of chlorophyll. And then you see what we call the near infrared plateau. That's from about 700 to say about 1350 nanometers. And this plateau is largely because of this near infrared scatter due to the canopy. And you can see some nice deep valleys. These absorption features are due mostly to moisture, leaf or canopy moisture, sometimes soil moisture. And there's also the difference between the visible and the near infrared is this red edge. And the red edge shifts depending on how stressed the plant is. Blue shifts when it's not stressed, and red shifts more to the right when it's stressed. And that can get at stuff like leaf nitrogen and just general, it's a general indicator of stress. Going further out into what we call the SWIR, shortwave infrared, these are very long, low energy wavelengths. The SWIR 1 and SWIR 2, we can see different features. And these are mostly associated with moisture or what we call non-pigment compounds. So if you're interested in dry residue management or detecting dry residue, this is the part of the spectrum you're interested in. And so you're picking up carbon compounds that aren't necessarily directly related to photosynthesis. So a lot of people, for example, use this, not only dry leaf matter, but also if you're wanting to assess the biomass of trees, tree trunks, these aren't photosynthesizing, but these are good indicators of carbon. And so you can imagine if you're looking at Landsat or Sentinel-2, you're only dealing with, 6, 7, maybe a dozen bands over this entire range. And so you're losing a lot of the information. Yeah. So I'm going to zoom in because some of you mentioned the accessory pigments and the visible range. And this is, just to give you an idea, thinking of other indices you might want to use for photosynthesis. So we're going to zoom in on the visible because this is, I would say, the most intimately connected to photosynthesis. So this is from about this chart here, 350 to 700 nanometers. So this is Roy G. Biv, if you know that anagram, red, orange, yellow, blue, indigo, violet. So this is blue, green, yellow, orange, and red. And basically, this shows absorption. This is a bit misleading. This is the opposite of reflectance. So for example, chlorophyll A absorbs very strongly in the blue, around 400 nanometers, and very strongly in the red, around 650 nanometers. So this is not reflectance, this is absorption. So in addition to chlorophyll A, which is the main pigment associated with chloroplast and photosynthesis, you have all of these accessory pigments like chlorophyll B, the carotenoids. The carotenoids reflect strongly, absorb strongly in the blue, but reflect strongly in the orange. That's why they appear orange. So carrots have a lot of carotenoids. Another example, if you've seen leaves senesce, this is a maple tree. As fall approaches, the leaves start to turn yellow and orange, and that's because the chlorophyll is gone, the chloroplasts are dead, and now all you're seeing is reflectance from the accessory pigments. The accessory pigments, in addition to increasing the amount of energy that's absorbed by the canopy, also help to dissipate when there's too much energy. And I showed this for any of you who've had a dead battery in your car, you had to get your car jumped. One of the things, if you don't know, they tell you to do is to turn on your lights before you jump the battery. And that's because when you jump the battery, you're sending a tremendous amount of energy into the engine. And so turning on the lights, turning on the air conditioning, turning on all the switches sort of helps to dissipate that energy. And that's exactly what these accessory pigments are doing. And we have indices. If you look on that Harris web page I showed you, indices that specifically address these different accessory pigments. Okay. Another question from Dr. Subash. These curves are supposedly for an ideal crop, genetic pattern for NDVI temperature. Not for an ideal crop, no. I think, Dr. Subash, you must come from the crop modeling community, like DSAT. Yeah, so you have to think of crop modeling in a bit of a different way. These factors are, you can consider them sort of first order factors or controls on crop yields. And we do that so that we can use remote sensing and we can do estimates over large areas. And so these are not ideal because, of course, the factors are for sub-ideal. Like I said earlier, the difference between potential versus actual yield, that yield gap. So these types of models can address that. But if you're looking for models that are looking at, say, irrigation timing or fertilizer application or other farm management decisions, very descriptive models, this is not the type of approach we use. And that's also because these types of models largely do not use remote sensing. If they do, they use what's called data assimilation. So maybe leaf area index estimated by remote sensing is used to correct leaf area index estimates in the crop model. But remote sensing is not used directly and is not widely used in this community. So that's, again, the motivation of this webinar, to focus on this approach, because it helps you to understand what crop yield is, what drives it, but also this is the main approach that remote sensing uses at the moment. And quite successfully, I would have to say, like, these models are not bad. I got some very good results running at a continental scale. I got an R² for soy and for corn and rice above 0.7 with district level crop yield data. So, and anybody who's worked with district level crop yield data knows it's not always the best data in the world, and I still got some pretty decent results. So, but yeah, that's another topic for another day, data assimilation. That's something worth exploring too, if you're really interested in sticking to these sort of very descriptive models. Okay, so putting it all together. So this is just one model, an example, but a lot of them look like this or variation. This is the production efficiency model optimized for crops, PEMOC. This is a model that I developed and published in RSE in 2018. And so you can see some of the constraints I talked about are not here, like the nutrient availability. So my model doesn't look at that, but that's there are a couple of reasons for that. One, the functions themselves are not that great, I don't think. And then the other restriction is data. So the idea is this can be run anywhere in the world. So the main input is PAR, the amount of light available for photosynthesis. A fraction of that is used to produce biomass. This is basically your utilizable PAR. Some people don't have this factor, some models do. So basically, I assume that all things being equal, that about 63% of the light available for photosynthesis is actually used to produce biomass. And then these other factors are included. So I have the quantum efficiency. This is the carbon part for C3 and C4 photosynthesis. So you can see for C4, I assume a constant. For C3, it then becomes a function of temperature. And that's because temperature is much more important in controlling the reactions in C3 photosynthesis. So this is a Collatz model, COLLATZ or Z. fPAR is another approach. But some models don't even have this. The Modis GPP product, for example, uses a lookup table. And then you have fPAR. In the case of this model, I use NDVI, but other models use EVI. Some use ratio-based approaches versus this linear approach that I show here. Leaf age, my model includes leaf age. A lot of models don't. A lot of models include diffuse light, or I should say not a lot, only one model that I know of includes diffuse light versus direct light. And this is basically calculated fPAR divided by fPAR max. And so that produces that chart that I showed you on the leaf age page, where basically leaf age is a function of fPAR divided by fPAR max, which occurs around maximum photosynthesis. So this starts affecting the relationship basically between NDVI and GPP later in the growing season. And then you have the moisture constraint, which in this case is based on vapor pressure deficit. And then finally, the temperature constraint. So if you want to see how these look visually, just go back to the previous slides, because basically I used these functions to define those images between FT, FM, FA, and GPP. Yeah, so this is a basic schematic of how these models are. Are there, I think this is a good time maybe to ask if there are any questions. Ooh, Dr. Subhash, man, I think you must be a hardcore DSAT or APSOM person. No, this is not, this is classified as a semi-empirical model. But if you think about it, process-based models like DSAT are also empirical, right? Some of the functions are physical. They use differential equations based on the physical understanding of the system, but a lot of them are empirical. So really, if you want to get down to it, all models, all process-based models to some extent, even global circulation models, GCMs, which are supposed to be the gold standard in process-based modeling, have empirical elements. But if you want to really distinguish, if you use the Lebel classification, the paper I provided from 2013, Lebel distinguishes between purely empirical, and I think what he's meaning there is machine learning, deep learning, regression, sort of these statistical techniques, semi-empirical like this one, which has some physical basis. So these relationships didn't come out of nowhere. These relationships are empirical, but they're based on extensive laboratory and field experiments. So, and they have a physical basis. So that's why they're not purely empirical. And then you have the fully mechanistic approaches like DSAT or APSOM or ERISA. These models are somewhat more physically based, right? They're certainly dynamic. They interact, they have feedbacks. So they're, more mechanistic or process-based. But in fact, those models also include empirical elements. But PEMOC is just an illustration. So the most common data you'll probably use in Africa are WAPOR or the DMP product or MODIS GPP, NPP. These products are all based on the big leaf approach. So it's a good one to understand if you're going to use if you're going to estimate crop yield going forward. Yeah? But good question, Dr. Subhash. Always good questions. All right. Okay, so here's an example. How can we calculate anonymous NP attendee? Oh, yeah, this is the point of the lecture or webinar. I'm sorry, you've sort of missed it. We calculate fPAR from a vegetation index or leaf area index. And that's shown here in this slide. This is just an example. So you see this function here, again, developed through empirical studies. This is the normalized difference vegetation index is detected from remote sensing. And it estimates fPAR. It's a statistical relationship. You can also estimate it from leaf area index using the Beer-Lambert law, as I showed earlier. You can use more sophisticated approaches, radiative transfer model approach through what's called model inversion. So basically, radiative transfer models, you feed in Sentinel-2 data or some other remote sensing data, and you get fPAR as an output. Most of the time, these models are run in what we call the forward mode. So you feed the models with fPAR and these other biophysical, biochemical parameters, and they estimate some kind of spectral profile. But in the inverted or backward mode, you get the opposite. So I don't go into that method because that's not the big leaf approach, and that's not widely used for crop yield estimation using remote sensing. But that's a good question. I hope you got it. If you didn't, I highly suggest you watch the video again and maybe read the reading material, because this is the essence of the talk. Okay, So here's an example of some light use efficiency models other than PEMOC. I just provide a list here for your information. I'm not trying to sell the PEMOC model. I'm not saying you should use it. It's not necessarily a good model, and it's not necessarily good for all conditions everywhere. It's just a way of teaching you about how these models are put together. So in fact, a lot of it, the model is loosely based on the CASA model. This is, I think, the first or one of the first, the Carnegie Ames Stanford approach, and MODIS GPP NPP. But the model is certainly better than MODIS GPP NPP. I actually discourage people from using the MODIS product if they can, because it has a lot of problems, especially for crops and grasses. That was the motivation, in fact, for creating the PEMOC model. You can see this one here, GPP VPM. This is a fairly recent model and publication. And then there's a few others here. You can see these, like the cloudiness index light use efficiency. This one's really good because it accounts for diffuse light conditions, which a lot of models don't. And in fact, diffuse light actually tends to lead to higher productivity, which is, yeah, you might not think is intuitive, but it's true. Copernicus, dry matter productivity, the DMP product. I don't know much about this product. I don't know how much it's been tested, but it's a blend between ProbaV and Sentinel-3. It's an ESA product, and it's available at 300 meter or 1 kilometer resolution. So you could also use this in combination with the harvest index to get at yield. Yeah. So the last set of factors which we don't address, and that's just because remote sensing based models don't usually address these, but to complete the story of the Jones paper, you should also know that there are reducing factors, and these factors also have an impact on yield. Unfortunately, maybe with hyperspectral remote sensing, you can get at some of these, but most remote sensing technology, yeah, good luck. You could also, if you're doing more precision agriculture using UAS or drones, you could get at these probably better with the higher spatial resolution. But the focus of this talk is on satellite-based remote sensing. So, and here's a few illustrations. Weeds, I'm sure know about. Usually we apply Roundup or some kind of herbicide to kill the weeds because they compete with the crops. Of course, there's a lot of debate around this. If some of you are familiar with permaculture, or you kind of let things stay as they are. But these can certainly be detected if the row spacing is significant and you have high spatial resolution imagery. With hyperspectral, and I should say hyperspectral is going through a bit of a renaissance. There wasn't hyperspectral information available from space for at least 10 years, maybe more. But now there are two new missions, one by the Italian Space Agency, PRISMA, and another by the German Space Agency called NMAP. NMAP, I know, is freely available. You can put in data requests, no problem. And these data provide pretty high spatial 30-meter resolution hyperspectral information, at least on a monthly basis. So in theory, with hyperspectral, you can better distinguish different crop types. I wouldn't say crop varieties. Some people say this, they try to sell that. I think that's not being honest, but certainly different crop types. And weeds would be included, so different vegetation species. You can certainly see with hyperspectral changes in the plant canopy due to pest and disease. There's a lot of work being done here. I give some examples here, aphids and blight, because these are, blight is a disease, aphids are a pest. These are two common things I have problems with. I grow a lot of my own food, and tomato blight, for example, spread because people planted potatoes everywhere for too long. If you're not careful, it rains here, the soil splashes onto the tomato and kills it within a couple of days. You don't have a tomato anymore. And then aphids, of course, Aphids are a huge problem. And they do permanent damage to the crop. So even after you get rid of the aphids, problem's still there. Anonymous attendee, are you aware of any studies done in countries like India or Africa where high heterogeneity in the crops in terms of using light use efficiency? Yeah, I can't point to any papers at the moment, but there's certainly studies done. At the moment, though, most of it's done either in China, Europe, or the United States. I think you'll find more studies in India and Africa dealing with these more sophisticated crop models. I have a student, for example, right now using SAFY, I think it's called SAFY, in Ethiopia. And he's doing data assimilation. So he's using leaf area index as detected with remote sensing to improve the parameterization of the model. So I think these kind of studies are more common in India and Africa. But it's not to say that you can't use these models. I myself have an ambition to do this. I just need money. You know, money's somebody gives me some money. So it's funny you ask, because I'm working on a proposal right now just to do this, which is to build light use efficiency based crop yield model for Africa. So yeah, fingers crossed I get the proposal. But good question. Okay. So now some other factors: air pollution, acid rain. Air pollution, certainly soot blocks out the sun, can affect how much light is available, can also clog the pores of the plants and affect production in that way. These problems are big where you have a lot of air pollution, say in India or China, for example. Acid rain, more of a problem in Europe, I would say, high acidity. So every few years, I notice in my garden, I have pretty bad years, crop failure. And a lot of that is due to the amount of acid in the water. And that's caused by pollution, but from more distant sources. Salinization, this is an issue. I show this here in the lower right-hand side. I'm from California originally. The Central Valley of California is dry, but you get a lot of sun. There's high soil fertility because of the alluvium, the mountains adjoining the valley, a lot of deposits. And so it's an ideal place to grow food, but of course there's no water. So the fields are irrigated, water is pumped, and as you might know, this increases the salinity of the soil. And so this white area here illustrated is probably what we call a dead zone. So this salt is on the surface of the soil, highly concentrated, and so almost nothing can grow there. And you can see here, maybe this is alfalfa. It's still growing there. Alfalfa is pretty hardy, but soon these fields will also be salinated. Yeah, so some of the problems with looking at these factors in a sort of big leaf approach is there's a large number and diversity of, say, weeds and pests and diseases. The relationships are nonlinear. The other relationships I showed are also nonlinear, but cannot be solved in a functional way. So I'm sure I haven't really studied it, but there's probably being work done to address reducing factors using machine learning or deep learning, because these modeling approaches account for nonlinear relationships and complex model structure. And then, of course, these things are highly variable in space and time. So you can see here, say, the salinity footprint. All of this soil probably has high salinity, but you can see it's only visible from the surface in certain patches. Also, similarly with pest or disease, one crop may be affected, even starting with one leaf, and then it spreads. So these are some of the reasons why we don't look at these factors. Okay, so I think this is kind of a wrap-up question. Why Do you think, what do you think are some limitations of light use efficiency on crop yield models? Yeah. So basically, we've talked about it already, but I just want to see if you've sort of absorbed the information. Again, I don't want to sell crop light use efficiency approaches. This is mainly just a conversation starter to get you, if you haven't had much exposure to this, to understand sort of the nuts and bolts of crop yield and how remote sensing is used. But maybe now in the question and answer, you can think of some problems we have with this approach. Alexander, you're from Ethiopia. Where are you from in Ethiopia? He's in Baridar. He's a student there, half-time, and a student here. He's also a lecturer in Baridar. Maybe you're lucky enough to live in Baridar. Baridar also has excellent beer, as you may know. Let's see. Alexander takes some time to respond. Let's see if he says anything. Okay. I can connect you. Alexander. Biniam. Okay. Oh, I get it. Okay. Alexander, you live in Israel. Okay, but you're from Ethiopia originally. Okay, great. Yeah, I can connect you for sure. Oh, I'd love to talk to you. There's nice history between Ethiopia and Israel too. Anyway, that's a different story. My mind detracts. Okay, and then anonymous attendee, how much ground truth is necessary for replicating this work for a region? In addition, what instruments are required? Okay, that's a good question. You could do it in a number of ways. So what I did is I used the eddy covariance flux tower data. This data are available globally. They measure changes in CO2 and moisture flux over a field. So it's essentially point data. There are hundreds of these towers. Yeah, so that has some limitations. And then basically I developed the models at that level. And then I sort of validate the models with district level crop yield data. But another way you could go possibly is to use, there are these websites emerging now. YEN, Y-E-N, is an example of crowdsourced crop yield information. So these are data that are provided by the farmers. So you can get field level estimates of crop yield. And it's crowdsourced and it's free to my knowledge. And so, yeah, you can get fairly decent spatial representation, maybe not so much in time. That's another source of data you could use. Yet another one, you see this now in a lot of papers, is to use harvester data. But the harvesters aren't necessarily good. I think they require a lot of calibration to be a very good indicator of crop yield. But basically harvesters, as they're harvesting a field, they actually estimate the crop yield. So a lot of people use this information because you can imagine harvesters in a lot of areas, certainly in Europe and the US, maybe not as much in Africa, but to some extent, an area grows sort of one main crop, like corn. And so maybe they're harvested about the same time. And so you're getting a fairly large spatial representation of yield. And that information probably has to be acquired from companies. And then yet another one, like my student in Ethiopia, what he does is he interviews the farmers. And the farmers, tell him how many bags of a particular crop are extracted from each field. And they sort of take an average of bags and come up with estimates of yield. And that's actually pretty accurate. And some might argue is even better than clear-cut samples, which is the traditional sort of sciencey way of estimating yield, where you go into the field and you cut and burn it and measure the dry weight. And that's because instead of getting, spot samples of yields in the field, you're getting yield over an entire field. And the farmers, contrary to what a lot of people say, the farmers know their stuff. And so the information tends to be pretty good. And he's gotten some excellent results just based on farmer surveys. So these are just a few of the ways that I'm aware of that you could sort of calibrate and validate the models. But the flux tower data, it's not essential, but it's nice because at the core of these models is basically GPP. Yeah, I hope that answers your question, anonymous attendee. Okay, so maybe if we can just in the last few minutes address this final question. What do you think some of the limitations of the light use efficiency approach, the big leaf approach to yield modeling are? Yes, anonymous attendee. Yen is crowdsourced. Outsourced in UK. It's mostly UK and some Europe. So if you're working in India or Africa, no. And that's something you might think about too in terms of a business model. You can get that data, but you have to be well connected to, say, seed companies, crop insurance, those kind of folks. This data is not easy to find. You can get, if you know the CGIAR, like CMIT, for example, they collect a lot of yield data, but I tend not to use these because they're usually collected under very controlled conditions. And I'm more interested in trying to estimate yield from fields where farmers work, like normal sort of growing conditions. But yeah, CMIT might expose it to like extreme heat or pests or like, and so you're not getting that sort of natural phenology. Okay. All right. Well, I think these are great questions. We've gone over a bit. So I'm just gonna advance to the final slide here. So these are some examples that I came up with of why the big leaf approach, just so you know, because I know some of you are like, this model looks a little, just so you know, there's a famous saying, there are no good models, but there are useful ones. And that's my philosophy, is I try to use models that help me to answer certain questions, but none of them are perfect or even close. So what are some of the problems? As we've pointed out, they don't account for pests and disease. They don't directly consider management practices like fertilizer or irrigation. These are the limiting factors. They do indirectly through fPAR. So let's say you don't apply fertilizer or you start skimping on the water, you will see a change in fPAR because of that. So that's why I mean it's not directly accounted for. Respiration is overly simplified. They do not account for different crop types or varieties. This is what Dr. Sabash pointed out. Big leaf models do not work well for complex canopies or landscapes. So, I mean, crops are fairly simple from a canopy structure perspective. You can understand like a tropical forest canopy, rainforest, for example, very complex. So these models have a lot of trouble there. Models are difficult to calibrate and validate. They do not account for soil moisture. That's changing now. Actually, I'm doing quite a bit of work in that direction with shortwave infrared and also with SAR to get at soil moisture, which I think is a better indicator of the moisture constraint on growth. Harvest index is sensitive to crop type and to the environment. So we assume that the harvest index is constant by crop type or the sort of climatic zone, but in fact it does change. And this is another active area of research that I'm working on to sort of use the biomass information to estimate the harvest index in an intelligent way, an automated way on a pixel basis. And then finally, models are difficult to scale and transfer. This is a problem with most models. I would say they're better than purely data-driven approaches and also fully process-based approaches. They're a compromise because they have some physical basis, which increases transferability. and they require fewer inputs. So I think that's sort of the main consensus summary of the sort of disadvantages of the approach. I hope I've covered also the advantages quite a bit. This is just an example of a fully process-based model, dissect, and I provide a citation here. And you can see, yeah, much more sophisticated. Weather, soil, genetics, pests, there's subroutines for all of this. Yeah, but you can imagine if you try to run this over a large area, there's a lot of complications. And this is my last slide, just a summary. Crop yield is a function of incoming solar radiation. This is based on Monteith, 1977. So that's the main assumption of the model. If there's more light available for photosynthesis, there will be higher biomass accumulation and ultimately yield. And the reason why this works with Earth observations, because that's exactly what it's doing, is detecting basically the interaction of light from the sun with the canopy, or emissions if you're talking about SAR. Other factors further constrain light use efficiency, so I hope I went into those. These are the CO2 is part of the potential factor, but temperature, water, humidity, these are limiting factors. These are the main constraints on light use efficiency. fPAR is kind of subsumed under the CO2 basically. fPAR is technically the main constraint, but these are other big factors affecting light use efficiency. And so the main assumption, and you could say limitation of big leaf approaches, is that they assume canopy light interactions scale linearly. So that's why it's best suited for sort of regional or global scale applications, poorly suited for heterogeneous canopies and probably field scale assessments. And non-biophysical biochemical factors are not directly considered. They're sort of subsumed under that fPAR, because fPAR responds to these changes. But if you say ran a sensitivity analysis, you're not going to easily find, oh, is it fertilizer that's limiting fPAR, or is it water? Or yeah, it's hard to do that with these types of models. But if you look, yeah, a lot of the sort of agricultural monitoring platforms that are actively being used use this kind of approach because it has certain advantages. So I think that's sort of the end of my presentation. I can try to, I think, yep, I've answered all the chat questions. Are there any other questions before we close the conversation? I was really impressed, really happy to see so many good questions and people following throughout the whole presentation. That was really nice. Questions? Anybody still here? Okay, I think a couple of you left your email addresses, and so I will email you the practical assignments. Abdullahi. And who else? Alexander. Alexander, I also need to connect with Biniam, I believe. Is that right? Yeah, okay. Oh, and Lucas, do you want the presentation? Or, I mean, the practical assignment, yeah? Yeah, I can forward you the practical assignment. No problem. But like I said, if you want to know more about the content presented here, we will have an online course, face-to-face course later this year, so you can learn more. Okay. Anyone else? It's now or never. Okay. Oh, And from Italy, Stefano. CNR. Oh, what are you doing here? No, joking. Okay, so is it possible to estimate? Oh, anonymous attendee. Sorry, I didn't catch this one. Is it possible to estimate maximum light use efficiency from remote sensing? Yes. So that's basically your potential, right? So a lot of models, yeah. So before you account for the constraints, basically that is your maximum light use efficiency. So given the amount of carbon, the amount of PAR, then you estimate your potential or maximum light use efficiency. And then the constraints basically scale that down to get the attainable yield or biomass accumulation. Yeah? Edward, okay. A lot of email addresses. Okay. Yeah. Edward, but Edward, you didn't provide, oh, you did you? Yeah, okay. Yeah, I'll send you the practical assignment. So basically it uses Sentinel-2 data, Sentinel-2 data and I believe crop biomass or crop yield in a study area in Italy. Yeah. Okay. Well, I guess with that, I'll close the presentation. Thank you all for attending. I hope it was useful for you and you learned something. Somebody had a question, but Fatima, I would say, but let's see. Do you have a question you want to ask? Are you still here? I don't know when you raised your hand. It might have been a while ago. Do you have a question? Are you still here?
Hey.
Hi.
Yes, thank you for the presentation. Yeah, I raised my hand since a while, but I think, yeah, most of the things were so clear. And I would like to have access to the presentation, please.
Yeah,
Yes.
okay, great. Yeah, I'll talk to Yob. He's our technician helping us with this. I don't know exactly how it can be shared. Hopefully it can easily in Zoom once the presentation is done. I think either he or I will be given the option to provide it. So yeah, it should be shared to you. But please do, did you see the link? I put it in the Q&A. There is a recording of the webinar. It's not as extensive as this presentation. It was recorded before, but if you want to use that one as well, it's there. Did you see that one?
I don't see the link, but I think the link should be enough, yeah, for me to go back to the presentation and have time to look all in detail.
Okay, sounds
Yeah.
good. Yeah.
Yes. Thank you
Okay.
so much.
Yeah, thank you. And thank you, everyone. Nice meeting you, and have a great day or evening or morning, depending on where you are in the world. Take care. Bye-bye.